Hello everybody, I'm Louisa Diva and welcome to the return of the Photosynthesis 2030 webinar series. These webinars are funded through the Capitalise project, which is an EU-funded programme exploiting biodiversity to boost the efficiency of photosynthesis for better crops. And the aim of these webinars is to promote communication and collaboration across the photosynthesis community towards this common goal of crop improvement. I'm delighted today to welcome my speakers, Professor Steve Long and Professor Dario Leister. Our first speaker today is Steve Long. He's a global leader in photosynthesis research. He's the director of RIPE, and his expertise in silico crop design and field analysis and interest in global atmospheric change has driven much work towards engineering improved photosynthesis for more efficient plants with improved yield that can mitigate climate change impacts. He was instrumental in the development of SoyPhase to evaluate the impact of future climatic conditions in crops, and has given invited briefings on bioenergy, climate change impacts and food security to President Bush at the White House, to the Vatican and to Bill Gates, and holds the Inkenbury Endowed Chair of Plant Biology and Crop Science at the University of Illinois and is a distinguished professor of crop science at Lancaster University. Thank you, Steve.
So I'd like to tell you about our work on enhancing and adapting photosynthesis for food security under climate change. The work I'm going to be discussing has been funded largely by the Bill and Melinda Gates Foundation, the Foundation for Food and Agricultural Research, and the UK Foreign Commonwealth and Development Office. I'm dividing this into three parts: why more yield, why photosynthesis, and what difference could it make? And then I'll just tell you about two of the things we've been working on in the RIPE project. So why? If we look at the situation in terms of the number of people who have inadequate food, i.e. they are short of calories, that number has been rising since 2014. Each year since then, the number of people who are facing hunger has risen pretty steadily. In 2022, it was about 800 million people, which is roughly 1 in 10 people are calorie insufficient today. Most of those are in South Asia and in sub-Saharan Africa. If we project forward, though, we can see that this problem is likely to become exacerbated. If we look at average rice yields across the world, then since 1960, they've been steadily rising because of improved genetics, improved agronomy, and so on. And if we think that that could carry on, by 2050, we would end up somewhere like this. However, the UNFAO project that because of rising world population and particularly increasing demand, especially from the more recently emerged economies, that this is the projection we need to be on. So what this suggests is that we're going to have an ever-increasing gap between supply and demand, which is going to force prices up, which of course will impact the poorest in the world most. And this is the similar trend for wheat. We can see this for soybean and for maize as well. Now, it's been suggested, well, we can solve this problem by changing diets. So if we have more vegetarian, if we eat less meat and dairy, there would be more grain available for everyone. And also if we got rid of wastage in the supply chains, then these would both decrease the amount of demand going forward. And that would be great if that occurred. But if we look at trends over the last 10 years, globally, in fact, they've been going in the opposite direction. Secondly, we would say, well, if these things do rather provide the solution going forward, that increasing the amount we can get per hectare of land that we're using is still a good thing, because that would release more land from agriculture for rewilding, for other uses and so on going forward. So increasing productivity per hectare of land we're using is going to be good whether we need it for food supply or not. And then, of course, we have the elephant in the room, climate change. Now, if we look at China, India, and Indonesia, these are the three biggest producers of rice in the world. If we look at the 70s and 80s, the years of the Green Revolution, per decade, they were increasing average yields per hectare by 30 to 40%. If we look at China, by this century, that's come down to single figures. And what is so significant is that China is investing so much more today in improving rice than it was in the 70s and 80s. This is telling us it's getting harder and harder to get these increases. So where does photosynthesis come in? Well, the Green Revolution and a lot of what's followed is about increasing genetic yield potential, i.e. creating crop seed which has the potential to produce more. So genetic yield potential is the yield we can obtain for a given genotype on a piece of land if it has adequate water and nutrients and we defend it from pests and diseases. In that situation, what determines yield is then how much sunlight energy is available over the growing season, how good the crop is at intercepting that solar radiation, how good is it at converting that solar radiation into biomass, and how good is it at then partitioning that biomass into the part of the crop we care about. So these three conversion factors really determine genetic yield potential. Now, if we look at the values of these pre-Green Revolution, and we have here what we think is the theoretical maximum. So for example, if we take partitioning efficiency, before the Green Revolution, our rice and wheat cultivars partitioned about 30% of their shoot biomass into grain. After the Green Revolution, these numbers became more like 60, even 65%. We don't think there's much headroom left if we're still going to have some stem, ear, etc. to support that grain. Similarly, post-Green Revolution, for our major crops, interception efficiency, how quick the crop is to get out of the ground, etc., has improved. But conversion efficiency has barely changed. Now, I've put this on the basis of a theoretical maximum. So what is that theoretical maximum? Well, if we work through every step of the photosynthetic process, from say 1,000 kilojoules of sunlight energy hitting the leaf to what we get at the end, so looking at the thermodynamics of every step, what we could say is that for a C3 plant, we should end up with about 46 kilojoules. So a maximum efficiency of about 4.6%. Now that sounds low, but in fact, our very best crops at best achieve a third of this, and most a tenth. So this tells us with photosynthesis, there is a lot of headroom there if we can find a way to exploit it. And the plant breeder and physiologist, Lloyd Evans, at his address to the Royal Society a quarter of a century ago, pointed out that it's just not apparent how we can double yield potential, because that was what would be forecasted as needed by the middle of the 21st century from what we're doing now, unless we can genetically engineer photosynthesis. But he was very skeptical that engineering of photosynthesis would be possible. Now, why do we think that's possible today? Well, photosynthesis is the best known of all plant processes. We know every step in the process. We know pretty well all the genes involved. We know the properties of most of the proteins, We even have the structures. We have kinetic constants for most of the enzymes. This allows us with high performance computing to simulate the whole process in silico and probe it in silico. And then, of course, crop genomes have been intensively revealed now, and crop engineering is becoming ever more efficient. So, but if we look at what was improved in the Green Revolution, these were 2 or 3 genes really, mainly affecting harvest index, also interception efficiency. But if we look at photosynthesis, we've got about 160 steps, all quantitatively variable. That gives you really an infinite number of permutations. Now, one way to tackle this is to use systems analysis. This is, of course, a well-developed approach to looking at production, for example, of cars. You know, if you've got this number of workers by resources, where do you put them on the production line to have the maximum benefit? And that is often done through computational analysis. So we set about doing this with the whole photosynthetic process, and this started really over 20 years ago. And Xingwan Zhu, who's now a professor of the Chinese Academy of Sciences in Shanghai, at the time joined me as a graduate student, and we set about starting this. So it took about 3 years to describe every one of these steps as a differential equation. Many of the enzymes, Michaelis-Menten, and not all of them, of course, obey that, but you know, that each one was coded so that we had this complete system. We then went about numerically integrating it, and the system crashed. But we were lucky to meet a mathematician, Eric de Sturla, at our computer center, who said, oh, I had exactly the same problem, and this is how I solved it. So we worked with Eric, and his solution worked for us. Then we asked him, well, what are you doing? He said, oh, I'm balancing rocket motors. So don't let anybody tell you that photosynthesis isn't rocket science. He did actually say that what we were looking at was more complex. And so he carried on working with us because given that this was a natural process, he knew it had to have a solution. Now, once you can do that, you can then start applying optimization routines to then say, well, if we've got this amount of resource in the proteins, where should they be placed to get maximum benefit? Now, of course, the other complication of this is that the environment of the leaf isn't constant. It's changing over the diurnal course, and it's changing over the growth of the crop. So this also has to be taken into account. And with this modeling, we came up after a while with, in theory, several ways in which we can improve photosynthesis, in theory, that is, from the models. And we also looked at, well, how complicated would that be? So to convert C3 to C4, we thought, you know, this involves a huge number of genes. That would be long-term, maybe requiring 15 to 20 years. But others might be possible within a 5-year framework. And we were lucky at this stage that the Gates Foundation started reading what we were doing and decided, essentially said, well, how about we put our money where your mouth is and you show us this can be done. The other criticism really that this modeling had was many people said, well, increasing photosynthesis is not going to increase crop yield because sink limitation is the big thing. Now, one of the other things I was able to do when I first moved to Illinois was to build this facility that we call Soybean. It's basically using free air carbon dioxide enrichment so that in these rings, the CO2 level is raised to about 600 parts per million, where ambient is about 400. And what that does with a C3 crop is to boost photosynthesis, because CO2 is a limiting substrate. It accelerates the rate of carboxylation, a Rubisco, and it partially inhibits oxygenation. So in these soybean plants, when you measure within here, you're raising photosynthesis by about 20%. And what we found was with soybean, that raised yield about 16%, rice about 12%, wheat about 14%, 15% rather. What we've also seen subsequently is actually in some cultivars, the response is much larger. And these are more modern cultivars where we assume breeders have actually increased sink size. Now, of course, these model predictions are great, but now when you're challenged with actually doing it and showing that this works in a muddy field, it doesn't look quite so pretty. But with the Gates Foundation support, we decided there were 7 different ways in which we might be able to advance this. And so what we set about doing was using our modeling to guide potential improvements. But what the Gates Foundation really wants is for these to go into crops, particularly of importance for sub-Saharan Africa, such as cowpea, soybean, and cassava, and also crops important in South Asia, such as rice. So what I want to tell you about is kind of 2 successes we've had. One is with upregulation of C-diheptulose 1,7-bisphosphatase. And the reason for this was the modeling. When we looked at the black is what the leaf has in terms of protein levels. The green is what the computer says would be optimal. And this one really stood out because on a logarithmic, this is on a logarithmic scale, the computation analysis was saying, well, there should be 7 times more of this enzyme. Now, our colleague Christine Rains at Essex upregulated this in tobacco, and here are 3 separate events with upregulating SBPAs. Plants are clearly more productive in the greenhouse than the wild type. So we wondered, well, why is this? Why hasn't it already happened? So we went back to our modeling, and then the suggestion we had was, well, the crop ancestor probably evolved in a CO2 concentration of half of what it is today. So we reran the model at 220 parts per million. That's the average for the last 25 million years. And what we found was this enzyme had no control at that level, but when you got to future CO2, it went up. And when we go to where we think it'll be later this century, it went up even more. Well, this was something we could test. Should say it also rose with temperature. And indeed, David Rosenthal, a postdoc, tested this in our soy face, and sure enough, at elevated CO2, the response to upregulation of SBPAs was even greater. Then we also added into this facility elevated temperature using radiators. That elevated the temperature by 3. And we found that relative to the controls, upregulation of SBPAs gave us an even bigger difference with the control. So now I want to go on to another manipulation that we've been making, which concerns non-photochemical quenching. Now, a leaf at the top of the canopy, which is not in the shadow of anything else, on a clear sky day, will see light in a bell-shaped curve. But if you go 1 layer down, what is happening is that leaf is going in and out of shade all day long. So when it goes into the shade of another leaf, the light level is cut by about 90%. And so what happens in a canopy when you see this drop is that, of course, photosynthesis drops, but it drops far more than it needs to. And it takes some time to recover. And if you look at this over in a crop canopy over the course of a day, this can amount to 20 to 40% of potential carbon assimilation if you could delete this lag. So what's happening here is that in full sunlight, chlorophyll is becoming excited. It relaxes by driving photosynthesis and fluorescence, but that's insufficient to remove all of that excited chlorophyll. If it remains in singlet state, it will then move on to triplet state, which can react with molecular oxygen to produce oxidizing radicals. So of course, evolution has dealt with this by developing a process, non-photochemical quenching, which gets rid of the excess energy as heat. The problem is that when a leaf goes into shade, it carries on losing a large amount of the excitation energy as heat when it could now all be used in photosynthesis. And it takes many minutes before that relaxes. So what is going on here in particular is that violazanthin is being converted to zeaxanthin, which is closely associated with this quenching process. When it goes into the shade, the zeaxanthin reverts to violazanthin, but these two are being interchanged all the time. So we reason from our modeling and knowledge from Arabidopsis that we need to upregulate the genes which convert zeaxanthin to violazanthin and violazanthin to zeaxanthin, and also a third gene, PSBS, which affects the level of attenuation of NPQ. So our colleagues, Chris Naogi and Laurie Leonelli, developed a construct from Arabidopsis that we would then put into tobacco to upregulate these three genes. And Kasia Glauacker and Vanna Kromdijk were the postdocs who did this in my lab. Now, one of the advantages we have with this is that what we were looking for were plants which showed the same level of NPQ in full light, but relaxed that NPQ more rapidly when we went from sun to shade. So we could pick that up through modulated fluorescence imaging, and we found three events in particular which showed a very even expression of these three genes. So we then, when we looked at them physiologically, for example, look at CO2 fixation, they were in this transition, they were fixing about twice as much CO2 as the wild type. These plants look bigger in the glasshouse. We could also see that in fluctuating light, they had a higher efficiency, but it was unaffected in steady state. And when we went to the field, we got on average about a 20% improvement in productivity. And we were excited that Science were interested in putting this on the front cover, although I guess because Science doesn't often cover crop experiments, they put a soybean plant on the front, which was prescient because that was what we were thinking of doing next. That, of course, took much longer because soybean, when you transform, it doesn't generate many seeds, requires a few generations before you can go to a field trial. But when we did go to a field trial, these plants were showing faster relaxation. They were showing similar levels of NPQ in full sunlight. But most importantly, the yield of 5 of these 8 events was significantly higher than the wild type. And these were all ones which were showing strong overexpression of our genes. And very pleasingly, when we looked at protein contents, even though we got more than 20% more seed, the protein content was virtually unaffected. and similarly the oil content as well. Now, the soybean, in line with agronomic practice, is not fertilized. It has to fix its own nitrogen. So the only way we could explain this is that the plants were using some of the extra photosynthate to feed the Bradley rhizobium, which is fixing the nitrogen. And this time, they got the right plant on the cover of Science. Now, Amanda also took this the next year to our soy phase facility because we were wondering, well, if you boost photosynthesis transgenically and then you add on to that a further boost through elevated CO2, will you see anything? Can the plant still make more yield? And in fact, indeed it did, that we're getting more seed here. At least it's significant at probability level of 10%, significantly different. Harvest index also went up slightly as well. So that was pleasing to see. We've now trialing this at a number of sites. This is the one on our farm in Illinois, but it's being tested at 10 other sites as well. This is now being taken on by Gates Agricultural Innovations, whose role it is to now get these innovations into soybean cultivars suitable for Africa and also into cowpea. And we're working with TJ Higgins, who is the person who has spent 20 years getting BT cowpea into West Africa. And 2 years ago, that was approved for farm use, and it's become very popular. And TJ is now working on putting our traits into cowpea, and here is initial trials in Puerto Rico. So if you want to know more about what we're doing, we have a website. And I should also say that I've been describing the work of a very large team, and this is an incomplete list of credits. So thank you for your attention.
Thank you, Steve. That was great. Really enjoyed that talk. The questions are coming in, but I think I get the first choice of questions. My question is, what would you say were the biggest obstacles for translation of your findings to breeders? So, you know, as you say, we have this huge wealth of plant science knowledge now that we never had before. But what is the blockage? What is like when you think of the COVID sort of problems in healthcare, translation seems to occur quite fast with the vaccine, but what can we do to what's holding up translation in plant science in your opinion?
Well, I guess I'd say two things from a scientific viewpoint. That I guess what we didn't realize before we started was, for example, it took us 60 events with tobacco to get those 3 that actually worked, because you know, we needed this balanced expression. And what we didn't realize was, depending on where the construct goes in the genome, you know, you get one event where one gene's expressing, you know, you get another where they're completely unbalanced. So really being able to target our constructs to a point in the genome where you'll get even expression is really important. The other barrier, of course, which is very well known, is that there is, of course, especially in Europe, huge opposition to genetically modified plants. So the framework you have to work through is very expensive, you know, probably far more expensive than creating these plants. And that is, you know, you've got to do all this testing, you've got to prove that the proteins are not in any way poisonous, even if they're proteins that are very well known, or there are proteins the plant already has. So the regulatory framework is a very big barrier. I mean, that has been lessened in the Americas, and we're beginning to see signs of it being lessened in Africa as well. but it doesn't seem to be changing very much in Europe.
Thank you. I have some more questions. Stomatal aperture and response is linked with the zeaxanthin content in the guard cell. So whether change in the zeaxanthin content also influenced the stomatal regulation in your studies, what is your opinion in this?
Well, we haven't looked at what would happen if we just influence the zeaxanthin content. But one of the things we did do looking at individual genes was we found that upregulating PSPS alone did actually affect stomatal conductance in quite a good way, because we actually found that we could decrease stomatal conductance by about 15% without getting a loss of photosynthesis, but decreasing water use by about 15% of the leaf long. So we are exploring that now to see, does this also result in a water use efficiency gain out there?
Okay. There's another one. Steve, how do you explain the slight lack of correlation between protein levels and yield increase in your photoprotection soybean plants?
Well, of course, this is a huge, we certainly do see a correlation there, but I think what is, certainly the best events are the ones which were expressing all three strongly. But what is really important is that these are balanced, and that is more important than the amount, because if, for example, you express more VDE than Z, what you're doing there is you're taking away protection. And in those plants, we can see more photo damage. The other thing we can see is if we get more VDE and not so much Z, we actually end up raising MPQ, lowers photosynthesis in full sunlight. So the balance is really important.
Okay, I have one more, and then I think we have to move on. The question is, did you test or think that wind speed loss, photosynthesis, or the internal evidence of photosynthesis?
Well, wind is certainly something which affects light fluctuations, although those tend to be so high frequency that we doubt that what we're doing will have much effect on those. Whereas we think particularly the passage of the sun across the sky is the one which is producing the frequency, which is most affected by what we're doing. Yeah. The internal CO2 concentration was not affected very much by this.
Thank you very much. Okay, so our second speaker today is Professor Dario Leister. He's held a chair of plant molecular biology at Ludwig Maximilian's University Munich. From 2013 to 2016, he was founding director of the Copenhagen Plant Centre, and since 2020, he's acting as coordinating PI of the EU-funded Photo Redesign project, which aims to establish new model systems and toolkits for the next generation of photosynthesis researchers to create a novel enhanced type of photosynthesis, combining different natural and de novo designed photosynthesis modules. The title of his talk today is Enhancing Photosynthesis and How Can We Redesign the Light Reactions?
Welcome to my lecture on enhancing photosynthesis, or how can we redesign the light reactions. And in the beginning, I like to acknowledge the funding. First, we have here the ERC, and secondly, this is a German consortium. It's Collaborative Research Center grant, and it's called TRR 175. Now, you have heard already by Steve that yield is a product of biomass and a factor which is called harvesting index. And if you consider biomass, and you see that biomass depends on the sunlight, which is used by plants, interception efficiency and conversion efficiency. And you have also heard by Steve that it's the conversion efficiency where there's probably ample room for improvement. And conversion efficiency is in principle nothing else than photosynthesis. You have also heard by Steve that photosynthesis is not a perfect machine. So from 100% of solar energy which receives a plant, under optimal conditions only 5 to 6% is used, depending on which plant species you look at. And very interesting losses occur at two places. One is the fact that all of our plants are green. That means they are not using green light. So more than 50% of the electromagnetic spectrum is not used by photosynthesis. And you also know about photorespiration, which goes back to the enzyme Rubisco, which is not perfect. In principle, from a biochemical point of view, it's even a very lousy enzyme, which is side reaction which we do not want. And for this reason here also we have massive losses if you consider here that only 6.5% of energy are left here, and we're losing like 6.1 before this. Now, why is this? Why is photosynthesis not perfect, as many people believe? Because evolution is believed to make everything perfect. Now, maybe evolution is just the reason why photosynthesis is not more efficient, because it was invented by cyanobacteria. And this happened in the absence of oxygen, because oxygen was produced by photosynthesis. And if cyanobacteria invented oxygenic photosynthesis, that means the photosynthesis which produces oxygen, then of course it was not used to oxygen in the environment. Secondly, cyanobacteria are living in the sea, and they are exposed to low light intensities. And during evolution, of course, lands invaded the surface, so they have been exposed to intense light intensities, 10 times, 50 times more than they are exposed in the sea. And of course, they are exposed to the oxygen, which has been produced during photosynthesis. And that might be the reason why plant photosynthesis is not as perfect as it could be, because it traces back to a photosynthesis which was designed, evolved during conditions which were very different from today. In fact, plant photosynthesis is associated with many regulatory and protective mechanisms, and they might actually compensate for the shortcomings of cyanobacterial type photosynthesis in present days. So how can we enhance photosynthesis? And I would like to mention 3 different ways. The first one is very straightforward. You try to use more light in the canopy of plants. So that means you make them slimmer or make them smaller leaves that more light can penetrate the canopy, or you make less chlorophyll, because it might be that you only need 50% of chlorophyll to make the same job. And having less chlorophyll means that more light can reach the bottom of a canopy. I would call this more light for each leaf. You have heard from Steve the VP set approach, which is in principle an approach to make plants better adapted, more fit to changes in the environment. So the VP set approach was designed to cope with the fluctuations in the incident light. But of course, we have much more abiotic and also biotic environmental changes and factors like temperature and drought, just as a few examples. And we all know that during climate change, these abiotic factors are becoming more and more detrimental to plant performance. The third approach is just making photosynthesis better. Better means that instead of this maximum of 5 or 6%, which actually is used for photosynthesis from all the light which could be used, we want to increase this fraction, which is then actually used for producing ATP and NADPH, and then of course to produce carbohydrates, proteins, lipids, and so on. And this means making the photosynthetic process per se better. Or I call this approach more photosynthesis per leaf. And this is what the talk will be about. In consequence, I guess that all these approaches might be combined at the end to make better plants for the next 20, 30 years. And it might look like science fiction, but let's imagine we are able to make the perfect photosynthesis. That means photosynthesis that can use all light wavelengths. That would imply that you have black leaves. And these black leaves might be so efficient that you need less of them, smaller of them, having a completely new architecture of very effective plants, which can be planted much more densely than before in the field. Okay, but coming now to the project. So the project is funded by the ERC. It's a synergy grant project, and this involves several groups. I want to highlight the contribution of Neil Hunter from Sheffield. He's a purple bacterial guy. This is Josef Komenda from Trebon in the Czech Republic. He's a cyanobacterial specialist, and myself, I'm coming more from the plant field. Okay, so what is the plan? The plan is to increase the efficiency of photosynthesis, and as said, this would have huge implications for mankind because it would really help to feed the world and to fight hunger. And our idea is to attack at this second part of the figure here. It's a 50% of energy which are outside of the photosynthetically active spectrum. And how can we do that? We can do that by using the natural diversity or variability of photosynthetic organisms. That means let's look at the extremes: eukaryotes, cyanobacteria, and purple bacteria, and you see that they absorb in very different regions of the spectrum of the light. So here eukaryotes absorb from 400 to 700, just the same area as we can see. That's the visible light, but they have this gap in the green, which makes them greenish. Cyanobacteria absorb less well in the very energy-rich regions, and they have less of a green gap, and that's the reason they are blue-green. And purple bacteria also have a gap here in the visible light, but they can instead absorb in the infrared. If you would be able to combine these features of all the three classes of photosynthetic organisms, we would have something which has probably, well, covered then 30 or 35% of the spectrum, which has not been used by any of the single organisms before. So that's what we call a black leaf concept, because such an organism would not be blue, green, or purple anymore, but black. So how are we going to do this? And I show you here already some more details on the photosynthetic machinery. So cyanobacteria here in the middle are the progenitor of chloroplasts, and for this reason, the structure of photosynthesis is very much conserved. So you see both have two photosystems, and they differ in peripheral parts, which are the antenna, absorbing light, capturing light, and these are the phycobilisomes in the case of cyanobacteria, PBS here, and the light harvesting complexes in the case of plants, which are not extrinsic, like here in the cyanobacteria. Extrinsic means they are on the membrane, but here in plants they are in the membrane. And if you go to purple bacteria, you will see that they are more different than the other two. So we have here a reaction center, which resembles in its basic features photosystem 2, but it cannot split oxygen. And we have also intrinsic antenna, which even have a similar name like the ones in plants, but they are very different because they take care that purple bacteria can absorb the infrared. So we want to use this natural diversity of photosynthesis, which is across kingdom of lives. And our idea is to combine this in one organisms, which would be Xenocystis, because it's a prokaryote. It's genetically can be well manipulated, and is also accessible to adaptive evolution approaches. Here you have more details on the photosynthetic machinery in the thylakoids of cyanobacteria like Synecocystis, and here a plant like Arabidopsis. Here you have Rhodobacter, which is more primordial, more simple than the other two systems. They all have a cytochrome complex, B6F in the case of cyanobacteria and plants, BC1 in the case of the purple bacterium, but still very much conserved. And everything which is conserved is depicted in white, while everything which is special and specific to the organism is depicted in color. So colorful are the antennae, so the complexes which absorb light and then transferred to the reaction centers. As said, we have the antenna of photosystem 2 and photosystem 1 in plants and green algae, which are in the membrane, which are good for high light intensities, containing chlorophyll A and chlorophyll B. We have here the phycobilisomes, which contains linear tetrapyroles. And here we have, again, the intrinsic antenna of the purple bacterium, which contains also bacteriochlorophyll to absorb in the far red region. So the idea sounds simple. Let's take all these different antennae, combine it into one chassis, which would be Synecocystis, and make a super photosynthesis. Another picture here, which shows you the features of the three antenna systems. So these are isolated antenna systems, and I had to look very hard to find these spectral curves in the literature. And you see already from these, if you combine the three antenna systems, here LHC2, antenna complex from plants, phycobilisome from cyanobacteria, and here the reaction center with the inner antenna from purple bacteria. If you could combine this would cover much more from the spectrum than each of the single ones. And these are only the isolated complexes. That means if you look at the entire machinery of the three organisms, this spectra would be even broader. So that's some of the plans that we have in the ERC Synergy Consortium. So we want to combine then different antenna and put it into one organism. So here, for instance, either natural LHC complexes or modified Belin antenna, which resemble the phycobilisomes, which are of course natural to cyanobacteria. And by this combination, of course, we can use more of the wavelengths. Another thing what we want to do is we want to insert the reaction center of purple bacteria into the cyanobacterium. This would become a third photosystem, photosystem 3, which would use then the infrared light and would make a kind of cyclic electron flow. So this are already two of the 5 work packages in this consortium. And the first work package is as a proof of concept, and also for long-term breeding approaches. We would like to transfer the photosystems from plants into cyanobacteria and then modify them in this system, which is much easier to genetically manipulate than, of course, a plant. A fourth work package is increasing light stress tolerance, and this will be also part of the talk. But you see, this is a very ambitious project. It covers synthetic biology, very extreme synthetic biology, and it covers also adaptive laboratory evolution in this work package number 4. And this requires, of course, a unique synergy between scientific themes, and for this reason, 3 different groups have teamed up for this approach. Okay, what are the problems of this? And is there hope that this is not science fiction, but realistic? So the bad news is it's not so easy as maybe described before. So if you want to exchange individual subunits between the photosystems, let's say between plants and cyanobacteria, then it's not so easy as believed, because although they're very similar in the amino acid sequence, so 90% sometimes and more, this is mostly related with a huge drop in photosynthetic efficiency. So actually the photosystems appear not to like to have foreign subunits, even if very highly conserved from other species. And this concerns photosystem 1 proteins, photosystem 2 proteins. And another example is light harvesting complex 2, which can be made only from one protein. And so far it was not possible to transfer this from plants into a cyanobacterium. Why is this? And the answer to this is the so-called frozen metabolic accident idea. And this means that photosystems and even their surroundings have co-evolved over millions of years. And therefore, because of the multiple relationships between the proteins, they're interacting with each other in the mature photosystem or during assembly, and there are multiple types of interactions and cofactors. So for this reason, they don't like to be exchanged, even if the other protein is maybe only 10% different. This is also the reason why suboptimal proteins became fixed. And suboptimal proteins would be, in this case, Rubisco. So Rubisco is also embedded into such a network of interactions, even if it's not part of a bigger complex, but a complex on its own. And also the D1 protein is believed to be such a suboptimal protein. So for this reason, it's really difficult to exchange individual proteins from a multi-protein complex. But what one can do is, of course, to exchange the entire complex. Given that the interactions of the entire complex with the surrounding is not as complex and intense as within the complex itself. Because we need to consider that it's not only the photosystem, which is needed to make a photosystem, but also their cofactors, pigments and other things, and assembly factors, which take care that the photosystem can be built up from individual subunits to something which contains then 30 or more subunits, like in the case of photosystem 2. So the lesson from this is single proteins are difficult to be exchanged, but if you want to exchange something, then do exchange everything. So this is another picture which shows this in a very rough way. So here you have a photosystem which is symbolized, and in the middle you have the proteins with the multiple membrane domains. In the periphery, more the things which have only 1 or 2 transmembrane domains, so less interactions with other proteins. And here these triangles represent peripheral proteins which bind to the photosystem on the membrane, and you see there are multiple interactions. If you exchange off the central parts, like shown here, which still is similar to the original one, it might still interact weakly with other subunits, with others not. And that's a catastrophe, and this might even be not compatible with photosynthesis. So what is the solution? Try to exchange everything, or at least the central part. And there's one example of such an exchange that has been done by Gimple and co-workers in 2015. And this is what I explain in the next slide. So what have they done? They looked at photosystem 2 in green algae, and they exchanged the core of photosystem 2, which has 6 subunits. And they did it by plasmid transformation. That means they made a synthetic chloroplast genome of Chlamydomonas, in which the distributed genes were all condensed into one region. And this region could then be exchanged by synthetic regions from other green algae. This synthetic chloroplast genome of Chlamydomonas could also serve as a control, because here you have still the 6 genes, which have been distributed before, but now they are in a different genetic context. And it's, of course, interesting to see how this relates then in photosynthesis compared to the wild chromosome. So what is the positive news? So you can go to this related green algae, which are shown here, and take all the six subunits for photosystem 2 and put it into Chlamydomonas, and what comes out is still viable. This is what you see here. So while the control in Chlamydomonas has it's probably a maximum maximum quantum yield of photosystem 2 of 0.66, or that's the effective quantum yield of photosystem 2. These 2 strains with the exchange in the 6 proteins in photosystem 2 are viable, but they have dramatically decreased effective quantum yield of photosystem 2. So it works, but not well. And if you compare now the synthetic chromosome in Chlamydomonas to the natural one, you even also have a drop from 0.77 to 0.66. So what does it tell us? It tells us in principle that this frozen metabolic accident idea is correct, so you can exchange probably more easily entire complexes than single subunits, but it might be difficult because you need to consider also the proper gene expression, which involves not only genes, but also regulatory regions, including promoters, terminators, and many other elements. And apparently, by making the synthetic chromosome in Chlamydomonas, you lost some of the regulatory elements. And this is the reason why this chromosome is not as effective as a natural one. Good. Another promising example is the story that you can express functional plant Rubisco in E. coli. And this is a direct confirmation of this idea of the metabolic accident scenario. So here you take both functional proteins, structural proteins, small and large subunit of Rubisco. You take all 5 chaperones or assembly factors. So if you know what is required to make Rubisco, and you put everything into an organism like E. coli, which has never seen photosynthesis in its entire evolutionary history, you can make functional Rubisco. So this is a proof that it works, and it also tells us that these 7 proteins are sufficient, necessary and sufficient to make Rubisco. Another example from our consortium is this experiments from Neil Hunter. He took the entire chlorophyll biosynthesis pathway, put it into E. coli, en route also discovering or confirming so far unknown enzymes, and he could produce a green E. coli which is able to produce chlorophyll A. An example from our consortium not published yet is that we try to transfer the entire plant photosystem into Synecocystis. And here we see a sketch how this works. So this is the natural situation in Synecocystis. These blue genes and operons represent the structural genes for photosystem 1. And in different steps, we have replaced these genes by the plant ones. And you see here, we have made a big operon of most of the plant genes. And we repaired some damage that we have done on the way. And this resulted then in a Synecocystis strain, which has no endogenous photosystem 1 genes anymore, but only the plant ones. And what I can tell you now is that this was not functional. So this did not produce photosystem 1. But when we made some tricky experiments involving adaptive laboratory evolution, we could reactivate photosynthesis. And it seems that this photosystem 1 is now somehow functional in Synecocystis. That's a long story, a complex story, and that's for another talk. So coming back to this nice picture. So you want to exchange dramatically a multiprotein complex or photosystem, then exchange the central parts of it, or ideally everything. You will have then weakened some other interactions, because everything is somehow interacting, of course, with other components of photosynthesis. And you might strengthen these interactions then by adaptive laboratory evolution, and then get something which is functional and maybe fully functional again. And this optimization, our experience is adaptive laboratory evolution, which works just fine. So take home from this part of the lecture is, do not exchange individual proteins, but do it right. So exchange entire photosynthetic modules. These modules must consider that also genetic elements for regulatory reasons might be required. That was the experience of the Gimpel et al. Experiment. And we might also need auxiliary factors like pigments or assembly factors. I think it's not too much to claim that eukaryotic photosynthetic modules can be swapped into prokaryotes photosynthetic microbes. So there are now literatures in the examples in the literature, like the entire chlorophyll biosynthesis pathway, or Rubisco, or what we have now in our lab and are trying to optimize this would be an entire photosystem from a plant in a cyanobacterium. In our opinion, the cyanobacterium Synecocystis is the ideal chassis for such an experiment, because it's still similar to plants, but it's genetically much more tractable and the ideal species for synthetic biology, and as you will hear in a second, for adaptive laboratory evolution. So let's assume that we are able to make a black cyanobacterium. How can we prevent that this is just burning out? Because imagine it can harvest light maybe with 50 or 100-fold increased efficiency. So how can this organism cope with this additional energy? And this second part also relates with the idea to make photosynthesis more robust, or to be able to cope with more light than it's naturally adapted to. So you know this curve, this is very traditional. You have the rate of photosynthesis depending on the light intensity, and theoretically it should be a linear correlation, but as you all know, too much light is harmful, even if it's a substrate of the process. So you run into photoinhibition and this kind of things, MPQ, we have heard about this before, and if you give too much light, then the organism will die. So the idea is, of course, to make organisms able to cope with more light, so they would survive longer and they could use much more of the light intensity. And that's the second goal of our consortium. We took Synecocystis, our chassis, for model experiments, and as you all know, the laboratory Synecocystis, which has been held for decades now in the lab, is adapted to low light intensities, which is different to the natural Synecocystis, which likes higher light intensities, but the one that we have in our labs is not surviving 1,100 microeinstein. It survives well at 700, and it really likes 100, for instance. So for all of you who are not familiar with this light intensities, the maximum light at the equator at lunchtime is about 2,000, and 1,000 is already a very nice sunny day, and 700 is what you have maybe in a place with nice artificial light or with some light from outside. So the lab type survives 700 but dies at 1,100. So what we have done is we have adapted the strain now to very high light intensities, exactly as what we expect would be the internal stress that the strains have if they would be able to absorb all wavelengths. And what you see here are adapted Synecocystis strains, which can survive 3,000 micromole photons per square meter per second, which is about 50% more than you have naturally on Earth. And this is not even the end of the story. This is just what we can produce. I'm sure we could also use 5,000 micromole, and they would still survive. So as comparison, you have here the lab type at 100 microeinstein, which is this color, and you see already that our adapted strains do not really look like Synecocystis anymore, but they more look like, I don't know, wet algae or lobster extracts. So how is this done? This is in principle very simple and straightforward. So what you do is you make evolution. You expose the strains to increasing light intensities over many generations, and to make this process even more efficient, you increase the mutation rate. And that have we done by applying either UV light or chemical MMS. In plants, you use EMS, but MMS works better for microbes. And we have done this now repeated times, and we end up with strains which have strange names like UMMM or UMUMM, and this means just that this strain, for instance, had 5 rounds of mutagenesis, UV, MMS, UV, 2 times MMS. And this strain then was first held at 700, which is good for the normal lab type, but the intensity of the light was increased during propagation up to 2,300 in this case. And then we took samples, so single clone descendants, which we sequenced. We also made sequences of the metagenomes during the procedure, and we ended up now with this adapted strains, and we ended up with sequences which allowed us to see which proteins have been mutated during this adaptive laboratory evolution. First question, of course, that you ask yourself is, okay, these highlight adapted strains might be highlight adapted and have maybe lost the ability to be good at low light. Actually, that is what we expected, and actually that is not true. So what you see here is the dry mass, so the biomass of the highlight adapted strains at highlight. There's a control, we have here the wild type at 100, and you see most of our strains, or these are batches of strains, are as good as the lab type under normal light conditions. So they're doing quite well under these enormous light intensities. At low light, also some of our batches are as good as the lab type. So what we can see is that there's no trade-off for being highlight tolerant in terms of being then less productive under very low light. It's probably interesting to know now for you which genes have been affected, and actually because we analyzed many different strains and also the metagenomes of the intermediate strains, we found hundreds of mutations in different proteins, and actually we found most mutations in proteins. And of the mutations in proteins, most were changing the amino acid composition. And several genes have been found independently in different strains. And these are the ones we focused on, which are shown also in part here in this table on the figure. And they are can be involved in many different things. So we found proteins involved in transcription. For example, here, this RNA polymerase subunit was found to be mutated at different positions in different strains. But you also have things which are involved in translation, or photosynthesis, metabolism, chemotaxis, and so on and so on. If I say we found in total 100 mutations, that doesn't mean that each individual strain has 100 different mutations to become highlight tolerant. That's not true. So you can calculate with about 5 or 6 mutations per individual strain, which has been necessary to make the strain highlight tolerant. But there seems to be a different way to make highlight tolerance. And for this reason, we found this many different mutations in many different proteins. So I don't want to go deep into this, but of course you can make a very detailed analysis of these mutations. So you can group these mutations into different haplotypes. If you look which mutations are in which strain, so you can also make a pedigree, how these mutations have arisen, and you see also the different phenotype of this single descendant clones. Here you have another evolutionary tree which tells you that there's a kind of epistasis in mutations. That means, for instance, if you make first mutation A, then maybe only mutation B can follow. While if you start with mutation alpha, then mutation beta might be the next ones. Interestingly, as I said, many, if not most mutations alter proteins and not promoter regions and other intergenic regions. Then we ask the question of how many mutations do you need in combination to make highlight tolerance? And we selected 2 mutations, which we found very interesting, and introduced them back into the wild type, into the lab type, which is not highlight tolerant. And I can summarize what you see here, which is a very crowded picture. What you can see here already only from looking at the cultures, that only introducing mutation 1 and 2 already is sufficient to make a wild type strain more highlight tolerant than without. So here at this light intensity, the lab type is dead, and this lab type with the 2 single mutations is still surviving. And of course, a deep physiological characterization can help you to understand why this happens. So what we're looking at is the OD, the dry mass, the pigment composition, the relative carotenoid content in terms of different components, and for instance, also the ratio of fucocyanin and chlorophyll. So this is a lot of data, and I'm not going into this. But 2 representative mutations alone can already enhance highlight tolerance. So you might need 5 or 6 to make it really extremely highlight tolerant, but already 1 or 2 are feasible to increase highlight tolerance. And of course, these are targets to be used maybe also in plants. Might be a long shot from the cyanobacterium to a plant, but it's a single mutation, and if the protein is conserved, very well conserved between cyanobacterium plants, it could be transferred also into plants. And actually, that's what we are trying in the moment. So the take-home message from this is enhancing light harvesting must go along with enhanced capacity to cope with additional energy influx. Or if you want to phrase it more popular, you say, if you want to pimp up your car by putting putting in a bigger motor, which is much more powerful, you need also to strengthen your chassis, otherwise it doesn't work. As a case of proof of principle, we showed that we can make synecdoche cystis more robust towards highlight by adaptive laboratory evolution. And this strain that we generated should be also then good for harvesting or for harboring the black leaf concept. So this strain should be already adapted to coping with more energy. And what we are doing now, and also planning for the future, is to use many other conditions than only highlight. And of course, we want to do the same experience in green algae, which are much more similar to plants than cyanobacteria. And they are equally suited for adaptive laboratory evolution because their generation time is not dissimilar. Some are even faster growing than synecdoche cystis. But of course, the genome analysis is much more difficult because the genomes are larger because they are eukaryotes.
So thank you all for attending. Thank you for all the questions and thank you for your patience. I look forward to seeing you all next month. Thank you, Steve. Thank you, Dario. Thank you, audience. Goodbye.